All right, hi everyone, we're gonna go ahead and get started. So it is great to see all of you here for our first in-person talk of the semester. For those of you who don't know me, I'm Rebecca. Me and Natasha are the grad student coordinators for the internal speakers this year. So hopefully you've gotten some emails from me at this point and we'll see more of each other. So it is my pleasure to introduce Dr. Matt Holding, who is a research fellow at the Life Sciences Institute just next door. Matt combines comparative genomics and proteomics with the field of natural history and evolutionary information to study how antagonistic coevolution between species generates diversity and novelty, specifically the weapons that species use to attack one another or defend themselves. So Matt got his master's at the California Polytechnic State University in 2011, where he studied physiological changes in the brains of navigating snakes. He then earned his PhD from Ohio State University in 2017, working with Michigan alum Lyle Gibbs to understand the phenotypic and population genetic factors contributing to coevolution between rattlesnake venoms and molecular venom resistance in squirrels. Then he did a shared postdoc between Clemson University and Florida State University until 2020, where he studied the evolution of venom trait complexity, fang morphology, and their relationship to diet diversity in rattlesnakes, partly funded by an NSF grant. He then worked remotely with Marjorie Maddock at the University of Nevada, Reno, where he used comparative genomics and transcriptomics to understand how gene family expansion contributes to toxin tolerance in herbivorous rodents. And in April of last year, he joined David Ginsburg Lab here at LSI at UMich, where he applies high throughput tools to characterize protein-protein interactions in viperid snakes and the rodent prey. So let's go ahead and welcome Matt for his talk.
Can everyone hear me? Good back there? Tom, you hear? All right. Should I stand up here? Okay, that feels weird. All right, well, thank you all so much for having me out. My name is Matt Holding, and I am a postdoc in the Life Science Institute over next door to EEB in the Mary Sue Coleman Hall. And I'm also, as Rebecca mentioned, an affiliate recently with the EEB department here. I am most interested in the traits that govern interactions between species, and particularly between predators and their prey, but any antagonistic interactions where one species is trying to attack another or defend itself from attack, like host-parasite interactions. We're all dealing with a lot of those right now, aren't we? So, oops, okay. So one observation we can make is that organisms and their traits are variably complex. Simple single-cellular organisms up to multicellular organisms, and the traits they have can be integrated or modular or composed of multiple components or few components. And Darwin made this observation early on when he invoked his tangled bank metaphor for the evolution of complex organisms in nature. And he said that, imagine a tangled riverbank. Birds in the bushes, roots of the plants moving down through the soil, worms and insects moving through the roots, all interacting with one another. And this put an early focus in evolutionary biology on how the interactions between species might select for differential levels of organismal complexity and the complexity of traits that organisms use to interact with one another. And an additional component layered on top of all this is that species are embedded, depending on where they are, in different tangled banks. Communities of species can vary in the strength and nature of coevolutionary selection between species, the diversity of species present in one place or another, and the stochastic or not so stochastic nature of the contribution of organisms from different parts of the tree of life existing in one community versus another community, the phylogenetic backbone that makes up communities. And so which of these factors are most important for the evolution of complex traits and species interactions? Many interactions, and the interactions that I'll focus on today, are mediated by molecules. We can think of toxins like those in, say, a poison ivy that cause you itch and worry and pain. We can think of maybe chemicals that mediate interactions between symbionts, like chemoattractants or pheromones that give signals about, say, a flower being ready to pollinate or a fruit being ready to eat. And we can think, similar to the way we think about toxins, about infection and resistance. So the multiple proteins on the cell surface that are involved, like serine proteases and ACE2 receptor, in the infectivity of a coronavirus, in this case on the right. And one thing that unites these is that these are often complex sets of molecular traits involved. We rarely see cases where there's just one toxin in, say, the venom of an animal or the poison of a dart frog. We see instead repeated examples where many molecules, either organic compounds or peptides and proteins, are involved in the production of toxins. And so I think that the idea that nature's red in tooth and claw is maybe a little redundant, because tooth and claw kind of do the same thing. They rip and they tear. We could say instead that nature's red in tooth and toxins. Things are often ripping and tearing and causing physical damage, but many other interactions are mediated by toxins, thousands of them. If you think about all the possible dyadic interactions between, say, the 15% of animal species that are venomous, we're talking about many millions of interacting species whose interactions are mediated by venoms. So the ubiquitous nature of these interactions alone means to understand biological evolution, we need to understand the evolution of venoms and resistance or susceptibility to venoms. Additionally, this can give us a guideline if we understand when complex, functionally distinct venoms are expected to evolve or not. We can maybe say where the best place to look for a new drug might be. So another simple way to put this is, as my PhD advisor Lyle Gibbs put it, why can't a snake just have one venom protein? Why do you need 20, 30, 100 venom proteins in the same animal just to kill a mouse? And so one emerging idea about the evolution of toxin complexity in these antagonistic systems is that toxin complexity is sort of beget or selected for by a need for functional diversity. So here's an early example from cone snail toxins, I guess a relatively recent example, not an early example, although in this field it's a relatively early example. The number of mature toxins across several cone snail species is positively related to Shannon's H index of diversity of species in the diet of those snails. So the idea here is that to eat a more diverse diet, you need a more diverse venom. A similar trend was seen in plant phytochemicals, defending them from herbivores. In the genus Piper, a diverse tropical genus of plants, when more insect herbivores were found on the leaves, there were more diverse sets of toxins in the leaves. So this idea of simple Shannon's H index or other sort of gamma diversity metrics of counting either the species interactions involved with a toxic species, either attacking or defending, sort of combines a lot of interesting components of biodiversity into one, and so in so doing masks several potential alternative hypotheses for the relationship between complex molecular traits and the communities of organisms with which species are interacting. And so today I'll talk about how snake venoms, as well as the P450 detox genes that rodents use to eat toxic plants, can tell us parallel stories and test alternative hypotheses about the relationship between molecular complexity in toxins or detox genes and community interactions in these species. So part one of this talk, I'll talk about adaptive variation in snake venoms, touch a little bit on work that I did in my PhD at The Ohio State University. In part two, I'll expand out to my joint postdoc work between Clemson and Florida State University, where I'll talk about the evolution of venom complexity across North American pit vipers. And then finally, I'll switch gears to talk about how we use comparative genomics and some dietary meta-analyses to relate detox gene diversity in myomorph rodents to the plants that they feed on. Okay, so I want to start with the idea of what is a venom. It's overused, but there's some good alliteration here. So venom is a complex cocktail. It's composed of proteins from several major unrelated gene families that combine in the venom gland to create this mixture that, when injected into prey, has this synergistic toxic effect that can do something fairly remarkable, completely incapacitate a mammal in about 10 to 20 seconds after the bite, so it doesn't get very far away from the snake. Additionally, venom is a cool trait to study because, for one, we can find a snake in the field or a scorpion in the field and lift the trait right out of it by a process that we affectionately refer to as milking the animal. You just take a venom sample, then you have your trait in hand in an Eppendorf tube. Additionally, because the final trait is a protein, we have a clear path from genotype to phenotype via the central dogma. And so we can link these proteins in a snake's venom directly back to the sequence variation that underlies that phenotype and its complexity. And additionally, and importantly, among species, even among populations, sometimes among individuals, venoms are highly variable, and we can test for the adaptive or potential adaptive significance of that variability. Oops. So here's just a genomic snapshot of an eastern diamondback rattlesnake genome that we're currently working on. And so each of these stick diagrams, I'm going to go through this fairly thoroughly because we're going to see some more of these later, is parts along a chromosome. And then we see regions highlighted and sort of zoom-ins on that region. And within the zoom-in areas, each of these yellow ticks is a single functional venom gene. And so what you can see, for example, in most of these cases, it's that there isn't just one SVMP, snake venom metalloprotease, making up the venom of this snake, but rather there are 23 of them, all tandemly arrayed in the genome. Snake venom serine proteases, same idea, 17 of them, and so on and so on. We have this really complex genomic underpinning, genome-wide in the snake, contributing to the mixture of proteins in the venom gland of the animal. Here's some work showing similar ideas. This is from Matt Giorgiani, working in Sean Carroll's lab. And they were specifically focused on venom metalloproteases, and they have this nice comparative figure that I think is really beautiful and shows this idea really well. From a single ADAM28 gene, a non-toxin metalloprotease that's present all the way back to the coelacanth here, or out to the coelacanth, you can see that in the king cobra, we have first 2 venom genes appearing, and then we have 30 of them in the western diamondback rattlesnake. But from additional recent work by my colleague Mark Margris on the tiger rattlesnake, we see that even though it's closely related, we get back down to 5 genes. Why so much dynamism? Why so much back and forth, birth, death? Why so much copy number variation in these venom genomes? And one idea that is gaining steam and support, and I think makes a lot of sense given what we know venom does, is that there are differentially susceptible prey in the environment. So over here on the right is an example from some late 80s work on California ground squirrels, a textbook example of evolved venom resistance. This is in a snake-free population here, and a little bit of injection of the same venom that was injected into this more resistant squirrel results in massive hemorrhage and death. Here we have very little hemorrhage, and this squirrel is going to be fine. Same species, about 100 kilometers between these two populations. So different species of prey can be differentially susceptible, not just due to this co-evolved resistance, which we think is a major source of selection, but also we should be aware due to just baseline physiological differences. Killing a frog is different potentially from killing a mammal because they're different, even due to just neutral evolution since the couple 100 million years ago when there was a common ancestor. So I studied this idea of co-evolved venom resistance, starting my career in venom world during my PhD. We used a coarse, but I would say effective measure of venom inhibition by taking serum samples from a ground squirrel, in this case, And then we incubate the venom with that ground squirrel serum and measure the enzymatic activity of venoms before and after incubation with the serum. And this gives us a percent inhibition value that we can use as a metric of resistance across populations and in reciprocal crossing studies to test for local adaptation. And what I found during this work was that actually the major effector or predictor of squirrel venom resistance was elevation of the site across these 12 sites. You see that squirrels from higher elevation areas, maybe under more stress or, I don't know, oxygen depletion, we armed wave about quite a few things in the paper, are less resistant. But once we control for that main effect of elevation, we see a classic signal of local adaptation. High elevation snakes did better against high elevation squirrels than low elevation snakes did, and we see the opposite with low elevation squirrels. And this was work that I did with, pictured up there, my PhD advisor, Lyle Gibbs. Also, while at Ohio State, I worked with two undergraduates, Abby Pimento and Blair Perry, to zoom out a little bit in phylogenetic space to the species level. And we did the same kind of idea, a reciprocal crossing study, in this case of two species of rattlesnake, timber rattlesnake, which feeds heavily on gray squirrels, and the northern Pacific rattlesnake, which eats California ground squirrels. And we found the opposite. We found this reciprocal pattern, but we found that the squirrels were the sort of adapted player in this case. Gray squirrels were better at inhibiting timber rattlesnake venom, their local predator, than they were at the allopatric venom, and vice versa. So once you get out to these phylogenetic scales where species have been in allopatry for a really long time and sort of evolving away from each other potentially while co-evolving with their local enemy, you see these sort of a different pattern emerge where there is sort of differential species susceptibility to venom. And so we have coevolution and potentially just evolved variation due to species being in allopatry as potential sources of diversifying selection over long evolutionary timescales for venoms and their function. And so this, for me, set the stage for the comparative questions that I'll talk about in the next part of the talk. And that's how and why does toxin complexity evolve among species over longer evolutionary timescales? And I did that as part of my work on the SCALES project, which was a joint Dimensions of Biodiversity NSF grant and Brazil FAPESP grant given to 3 institutions in the US, Lyle Gibbs at Ohio State, Chris Parkinson at Clemson, and Darren Rakita at Florida State, and then in Brazil to the Instituto Butantan, to Ana Moura da Silva, Felipe Graziatin, Erica Hinksahir, and Ignacio Azevedo. I also want to take the time to acknowledge that we did extensive collecting in Mexico during this work, and we probably would have found next to nothing without the help of our collaborators at UHED, Gama Gaitan and Miguel Borja, and also our collaborators at HERP-MX, Jason Jones and Chris Grunwald. Okay, so let's start with an observation, as we should in science. And in this case, one of variably complex venoms. So these are chromatography profiles of 2 species venom. We have a black-tailed rattlesnake on the top here, and a sidewinder on the bottom. And these chromatography profiles, this is time on the x-axis and absorbance at 220 nanometers on the y-axis. As protein passes under the detector, we get higher absorbance. So these peaks are going to be associated with the presence of a protein at that elution time, and the height of the peak is associated, with its relative abundance in the venom. So I hope it's obvious that this venom is much simpler than this venom. There are more components in this sidewinder venom, and they're more evenly distributed in terms of their relative abundance. Whereas this is dominated by essentially 2, maybe 3 venom proteins. So why? What are these phenotypic differences potentially conferring to the snake. And I'll posit three adaptive hypotheses in addition to neutral evolution. And we can differentiate, I think, between the four of them. First is the idea of pairwise coevolution. So what if we have a situation where tightly coevolving with maybe one or a few species generates that arms race, and the solution is to have many arms for your race? If you're tightly coevolving with one or two species under this hypothesis, we'd expect fewer prey species to select for more complex venoms. And so if we set up two hypothetical populations of snake, we would say in this case we'd expect or predict that this snake species over here would have the more complex venom. This one over here would have the more complex venom. It has fewer prey, and we might expect that to be a surrogate for tighter, more intense coevolution with just those two key prey. Whereas this one can kind of be more diffuse in its interactions with three main prey. The alternative then is diffuse coevolution. What matters is just the number of species, regardless of what part of the tree of life these species come from, that you interact with. It's sort of a one-for-one idea. For every prey, you need a venom protein. So under this hypothesis, we predict that more prey species select for more complex venoms, and that this snake species with three prey would have more complex venom than this species here. Okay, third, we could posit a phylogenetic diversity hypothesis, which might be expected to be a surrogate for functional diversity in the prey. So three species of prey in both hypothetical snake species here, but this species is eating all mammals: a rat, a rabbit, and a squirrel. This one is eating a squirrel, a European starling, and a lizard. Much more phylogenetic diversity present in the diet of that snake on the right side. And we'd expect under this hypothesis for it to have a more complex venom. So we set out to do field work. We collected, without much exception, every lineage of Agkistrodon, so the copperheads and cottonmouths and cantiles, and Cistrus and Crotalus, the rattlesnakes, present in North America. So in total, you're going to see a data set of 226 snakes from 68 lineages. And for each of these snakes, we would capture them in the field, take a venom sample, and then 4 days later, usually in some unfortunate hotel shower or lab in Mexico or the Southwest, we would dissect the snake. And this is going to result in a museum specimen that, depending on the location of collection, was either deposited in the US or in Mexico, and tissues which we subjected to RNA-seq. So we took venom glands from the snake, put those in RNA later. They bounced around in the field with us for a couple months, and then we did RNA-seq on them later. And so first, let's talk about the venom and the way we phenotyped it. I alluded to it earlier. This was with HPLC. So we get this series of peaks. So we get peak presence and peak relative abundance. And so with the area of the peak being the relative abundance data we used, we could then calculate Shannon's H, number of peaks and relative abundance, as a measure of venom complexity in the venom proteome. We also calculated venom sequence complexity from the transcriptome data. What we did here was we had a large database of venom coding sequences. We mapped our reads from each snake to them, allowing us to get rid of non-toxin reads in the venom gland. We used jellyfish to calculate the presence of all unique k-mers in the venom gland reads for venom genes specifically. And then we get also not just their presence of each unique k-mer, but relative abundance data again. So we can calculate Shannon's H of the sequence underlying each venom phenotype, a measure of sequence complexity of the venoms. The first thing we actually did with these data is use all of the non-toxin genes we assembled to create the first genome scale phylogeny, sort of a mini locus phylogeny for this group. So we can see that over here, sort of the beauty of the group, in my humble opinion, and I know some other folks out here might share that as well, snake fans. And we use this time-calibrated phylogeny for our comparative analysis to control for the similarity between related species in our attempt to assess the correlation between different metrics of diet diversity and different metrics of venom complexity. You can see our venom complexity metrics along the snake phylogeny here. In black, we have proteome complexity, and in gray, transcriptome complexity. It was comforting to us, given that we believe the central dogma that these were correlated metrics. And there was phylogenetic signal, which you can see as sort of the wavy motion, where more closely related species had similar venom complexities, but also plenty of variation across the tree to measure and explain. Here's this in a graph. More complex sequence leads to more complex venom-protein mixtures. And we have simple venoms with effectively 10 or so proteins in them, up to more complex venoms with effectively around 30 or more proteins in them. And so the next thing we did is leaned on the past century of really wonderful natural history studies, largely driven by museum collections of these snakes, to give us really solid metrics for diet diversity. If any of you followed Mike Grundler's work that recently came out, they used this exact same idea of mining the literature to characterize diet. In this case, we focused only on the species we were interested in. So we found 73 gut content studies that included 296 prey species for 30 of our snake lineages that we also had these venom complexity data for. And the cool thing with snake diet data, is that the whole prey item is in there. So you just pull it out, look at it. OK, that's a towhee. And in the papers, you'll see these nice tables that include the presence of all the prey species they found and how many. So we have presence and relative abundance data for each of our snakes that lets us calculate, as you can probably guess from how I've been talking about it, a Shannon's H index of diet diversity for each species. But we also wanted to add evolutionary information. So what you're seeing here is over 700 million years of snake chow evolution represented in a tree. This is a tree uniting all of the prey species that we found in those diet studies. And the tracks around the outside are representing the abundance of each species in these 3 example snakes' diet. So the timber rattlesnake had a really simple diet and a fairly simple venom. And so it's the outside track in blue, and you can see that most of its diet out here in blue are mammals. It's a mammal specialist. At the other extreme, the rock rattlesnake fed on centipedes, a couple lizards, a bird, and some mammals as well, spanning the entire tree. So we have an example where we can use this tree and the evolutionary information present to contextualize our diet information according to phylogeny. And that gave us a metric called mean phylogenetic diversity. So we have Shannon's H, just species diversity without caring about phylogenetic information, and then mean phylogenetic diversity, which recognizes that eating these 2 species is quite different from eating those 2 species. And so what we found was support for the phylogenetic diversity hypothesis for the evolution of more or less complex venoms. So species diversity alone, here on the x-axis, did not predict, in this case, our venom transcriptome complexity metric. But our standardized MPD, our mean phylogenetic diversity of the prey in each species diet, was positively correlated in a PGLS analysis with transcriptome complexity underlying the venoms. And the proteome complexity measures reflect the same trends. We can then, because these were transcript data, assign them to the different distinct and chromosomally distinct gene families that are underlying venom variation. And what we found was both support across 3 of the 4 gene families that we analyzed, but also evidence for heterogeneity across the families. So in metalloproteases, serine proteases, and phospholipases, we recapitulate the sort of venom-wide result. More phylogenetically diverse prey predict more complex expression of these venom gene families, likely more venom gene copies in these venom gene families. Not the case for C-type lectins. Why are they weird? Among the four up here, they're the only non-enzymes. They bind to their targets. They also act as heterodimers, sometimes up to octamers. So maybe they're constrained by the need to bind to one another before they actually enact their actions on prey. So now let's focus in on the serine proteases, because we know a lot about the targets of these. So this is some work from Solange Serrano in Brazil. And Serrano showed this nice figure of all the different places where we know about serine proteases that attack different members of the coagulation cascade. And so what I did was, from representative prey species across that prey phylogeny, I pulled down sequences from GenBank, amino acid sequences, of all these various coagulation factors that we know these serine proteases attack, and used the same mean phylogenetic diversity concept to calculate what I called a mean coagulation cascade distance. Same idea. Using the gene trees of factor V, prothrombin, kininogen, et cetera. And what we first found, comfortingly, was that more phylogenetically diverse diets had more phylogenetically diverse coagulation factors in them. Now, this is kind of a duh. We expected this, but it wasn't. This was actually a reviewer request, to be honest. And it proved to us that even in these highly conserved pathways, the coagulation cascade of vertebrates, more diverse diets reflected functional diversity in the targets. And in turn, this coagulation cascade distance predicted serine protease complexity in the venoms. We were to recapitulate those phylogenetic diversity relationships with just the distance between the known targets of those particular venom compounds. And so to wrap this part of the talk up, venom can either be simple and potentially prey-specific, or maybe complex and more functionally diverse. And this actually makes sense based on functional data from, again, folks at Instituto Butantan in Brazil. This is a South American species of lancehead snake, a Bothrops species, Bothrops nuidi. And these graphs are a reflection of the speed and final strength of clots that were formed in the blood of either a mouse or a chicken. And you can see that the whole venom had about the same effect in terms of the progression of a clot. But these are three different proteases from the venom tested individually. And you can see that two of them are pretty good at clotting bird blood. The third one kind of stinks at clotting bird blood, but it's the only one that clots mammal blood and clots it very effectively. And this species of snake eats both mammals and birds. So when we think about gene content in the chromosomes, these long tandem arrays of venom genes in snakes, the maintenance of them or the birth and death of them is likely a product, at least in part, of selection for sub or neofunctionalization to maintain a diverse diet in cases where snakes are feeding on phylogenetically diverse prey. And then other processes that we know are probably important here, like coevolution and drift, we expect to be sort of superimposed upon this sort of fundamental relationship of phylogenetic diversity of prey predicting venom complexity. It's not to say that coevolution is dead here. We know that snakes locally adapt to individual species of prey. It's just that at this scale, it's not providing us with a solid prediction. Okay, so I'm going to switch gears at this point and talk about what I'm affectionately referred to as my pandemic postdoc. I spent a year working remotely in Marjorie Matok's lab at University of Nevada, Reno. I was actually there for about 2 weeks before the pandemic hit. This work was a collaboration called the Genome's Underlying Toxin Tolerance Project, or GUT. And this is an EPSCoR Track 2 sort of foundation building grant that was given to several Western institutions. It's a collaboration between Denise Deering's group at University of Utah, Jim Henderson and Brian Simonson at Cal Academy, Jen Forby at Boise State, and then Marjorie, as well as Laura Richards and Tom Parchman at UNR. And so wood rats are interesting because they eat really nasty plants. They eat plants like Prunus fasciculata or desert almond in the case of the desert wood rat, Neotoma lepida, or Frangula californica, or California coffeeberry, in the case of its sister species that it regularly hybridizes with along these ecotones, the Bryant's wood rat. And so how do they do this? How do they maintain a diet that includes several things, but is pretty heavily weighted towards plants that produce really nasty chemicals? These toxic diets have physiological ramifications for these rats. These are serial urine samples taken by Danny Nielsen as part of his PhD work with Marjorie. We have some transcriptome data associated with this, but I won't steal his thunder today on that. I just wanted to show that the urine gets bloody the more of the native diet Bryant's wood rat ate. This is just increasing concentrations of its native plant diet rather than eating rodent chow, and blood in the urine. One way that we think that is crucial to solving these challenges for these wood rats are monooxygenases, the cytochrome P450s. So these inactivate drugs or xenobiotics via oxidation reactions that set them up to be cleared from the body. This is an ancient superfamily of genes. Most of them in vertebrates are single or double copy, sort of housekeeping or steroid producers, but 8 of them are called unstable families. So with Thanksgiving coming up, we can all relate to the idea of unstable families. But in this case, we're talking about families of genes that are tandemly arrayed, again, just like the venoms, and they're dynamic in their copy number evolution and also in their sequence evolution. And so this is some qPCR-based analysis of 2 species from Denise Deering's lab for P452Bs, which are thought to help a lot with terpenes and things like juniper. And there was evidence for multiple copies of these 2Bs and copy number variation between species. But we needed high-quality whole genomes to actually solve this. And so while working for Marjorie, I was involved in the production of 4 de novo high-quality genomes based in long reads for 4 species that all form a single symmetrically bifurcating clade within the genus Neotoma. And with these high-quality genomes, we were able to then manually clean up these P450 gene arrays and for the first time visualize the potential for copy number evolution. So sorry this is a little blurry, but this is where we expected to find these 2B genes in what's called the 2B-AGFST array. Those are just different letters representing different subfamilies. Each lettered subfamily is a different color. And so you can see really that what should stand out is there are 2 that are evolving kind of dynamically across our outgroup, Old World Rattus norvegicus, and then our in-group Peromyscus meniculatus, and then our 4 new wood rat genomes. It looked like most of the excitement was in the 2As, which we didn't expect, where the wood rats had increased their copy number of 2As to up to 13 functional copies, each of these small green arrows. Some kind of dynamic action going on in the 2Bs, especially with this dusky-footed wood rat, bumping it up to a number of 10, but that's not even much more than Rattus norvegicus. What's going on here? Where's all this special action that we expected? Well, it turned out that there'd been a downstream of about a 2 million base secondary duplication event that created an entirely novel gene island for the 2Bs, only in New World rodents. This is absent in the Old World rodents. Peromyscus has 2 of them. Bryant's wood rat has 2. And we get up to 29 functional copies of CYP2Bs in the array of the dusky-footed wood rat. We saw this repeated for several other cytochrome P450 gene families in these wood rats, creating pretty extreme copy number variation. And we wanted to put this, again, in the comparative framework. And as part of the EPSCoR grant, I was tasked with leading a genome annotation workshop that ended up turning into a real effort to bring in other high-quality rodent genomes and count cytochrome P450 gene copy number across several species and relate it to evolution of the diets of these rodents. And so what we saw was, in this case for another gene family, the evolution of 2 novel gene islands in first New World rodents, where island 1 appeared, and then secondarily, again, in wood rats and their relatives, an island 2 appearing. And we see this elevation in phenotype space in terms of copy number count. We then, again, in the same way that we did for the snake venom analyses, leveraged natural history information. We looked for studies of the diets of these rodents, calculated mean phylogenetic diversity of 460 million years of rodent chow evolution. All these are plants. And what we found was that for 3 of these unstable gene families, there was a significant positive relationship between eating more phylogenetically diverse sets of plants and the counts of cytochrome P450 genes in the genome of the 18 rodent species that we included in this analysis. And so if we look at the total count of these 8 unstable gene families in these rodents, we get a range of from 54 genes all the way up to more than 120 genes in one of our, this is Neotoma fusipes up here, and a significant positive relationship where plant phylogenetic diversity in the diet is able to explain 54% of the variation in gene copy number. So we see another case where variable complexity of a molecular trait, in this case antitoxin genes, was predicted by a set of sort of phylogenetically diverse antagonists. In this case, plants that didn't want these rodents to snack on them. So we also saw evidence that the formation of novel genomic locations, new gene islands, and potentially a new sort of background chromosomal context where these genes were occurring might be key to the more rapid acceleration of copy number evolution in these detox genes. And so our next steps here are to do some amino acid sequence diversity analyses at the active sites of these P450s and some 3D conformational diversity analyses as well. This active site shape and the expectation of being able to deal with different toxins vary when you have 40 2B P450s versus only 12 or 13 of them, like Gratis norvegicus does. Okay, some general takeaways from this work. First, in both of these cases, I had a lot of fun mining natural history information to be able to relate comparative genomic data back to what these organisms are actually doing. And for me, that's really important. I think I spend way too much time staring at the command line and not enough time staring at animals and plants. And so it was really great to be able to do that using literature information in this case, and information gleaned in many cases from museum studies. Species interactions have an important phylogenetic backbone. So the species that happen to be in a community and the part of the tree of life that they come from matters in terms of the future track that their evolutionary interactions might take. Oh, excuse me. And finally, novel ecological demands require complex phenotypic solutions. And last, I want to unite these parts of my work. It's always been important to me to sort of in parallel study predator and prey and know a lot about both and not just focus on one. Even though I started as a herpetologist, I've sort of become a bit of a mammologist, and uniting that through comparative genomics. And so we really need to know more about the molecular targets of venom in prey and the mechanisms, the specific proteins involved that confer venom resistance and allow, say, a ground squirrel to take more than 200 times the lethal dose that would kill a mouse and walk away with very little problem. But if maybe it gets 400 times the lethal dose, then it becomes dinner. And so that's actually what brought me here. So the sort of main part of the talk is over, and I just hope you'll indulge me for 2 slides to tell you why I'm here, and invite comments and thoughts should you ever have them as I'm continuing to develop my research program. I'm working in the Life Science Institute with David Ginsburg, who is an expert in blood coagulation and molecular genetics, mostly of humans and mice, but is interested more broadly in the regulatory mechanisms and genetic underpinnings of variation in many pathways, including the coagulation cascade. And so I'd approached him about potential collaborations involving snake venom because it targets the coagulation cascade. And sort of the rest is history, and I'm here now. And so one thing we're going to do is use a method that one of his postdocs, Laura Haynes, has really gotten going in the lab, random substrate phage display, which allows us to present all unique combinations of amino acids in 6-mers to, say, a venom, and ask questions like, do more complex venoms actually cleave more target proteins in a prey by mapping selected phage substrates back to the genomes of, say, a wild rodent? Laura, it turns out, is also an expert on a particular class of proteins that I've been previously interested in, serine protease inhibitors. So these proteins, whoops, it's not showing up. It's supposed to be a protein there. They have this loop sticking out from them that is a bait for proteases. And when a protease cuts that loop, they irreversibly bind it and inhibit it. And it turns out that, like many of these other stories that I've been telling, these serine proteases have shown massive copy number expansion in rodents relative to the single copy of some of these homologs that we see in humans. We also know from other work that's ongoing that these are under positive selection and that they bind to and inhibit snake venoms. So this is some of the types of questions and directions that I'll be exploring as I move forward here at UM, trying to understand complexity in the interactions between predators and prey. Thank you all so much for your attention. Any questions? Ivan, yeah.
So Matt, you had abundance data for detoxins and also the transcripts, right? What happens when you don't have abundance data? In a lot of cases, we just have presence and absence of composition data. Can you still make inferences similar to the
If you had what type of data?
abundance data, you just had like composition, like presence and absence of toxins, for example?
Yeah.
Have you explored that also in your analysis? How does that
So the raw numbers of genes we're working on a follow-up study are also predicted by phylogenetic diversity of prey. So in this case, just the numbers of transcripts, unique transcripts present. So it's not that, I don't mean to suggest that the relative abundance data is necessary. I think the number of components is certainly a dimension of trait complexity. We were just able to, we had both, so we were able to layer them and weave them both together. But I think it would be really interesting and useful to just count the number as well.
Sort of related to your comment about searching for the molecular targets of the toxins. Any comments on potential utility against pathogens?
Yeah, there have been a lot of explorations of venoms for use in drug development, not just for anti-pathogen work, but for the development of things like regulators of blood pressure and coagulation. Captopril is an example. There are insulin mimics, things like that have been used for treating diabetes. And there are antimicrobial peptides in venom, like defensins, for instance. Yes, I have access to libraries of snake venoms and absolutely no knowledge about how to test them for something like that. So please contact me if you have thoughts along those lines.
So I have a question about the stability of the diet. Say the two levels. One is that so for a given species of say snake, and you have multiple samples of that, multiple individuals of that species of snake, and you dissect, find the diet. Are they very similar or very different, right? And when you talk about the diversity of the diet, do you consider all those specimens or take average, or how do you
Yeah.
deal with that? The second question is the diet evolutionarily stable? Like, do you look at closely related species of snake, do they have similar diet? Because you try to correlate that with the genes that the snakes have. And the genes arise through millions of years of evolution. So if the diet has been changing very rapidly, then the correlation with the gene would be problematic, right?
Well, if I understood Mike Grundler and Dan Roboski's recent paper even moderately well, which is maybe a good question there, but a lot of the diet diversification happened early on in mammals, and then in snakes, and then there's been subsequent radiation to new areas of dietary niche space. Now, what I can say specific to these data is that even across space in the same species of snake, we find variation. I mean, some snakes range some snakes are very local in their distribution. Others range from, Canada down in south of the border with Mexico. And so their diets certainly vary over that latitudinal gradient. So in some ways, maybe it surprised us that we found this relationship, which suggests it's actually that there really, we took that as evidence that as a glass half full, there really is something here. And if we were able to better control for local variation, we might even see a stronger pattern. There are circulating haplotypes of venom genes within some of these species. So there's local variation or spatial variation in the gene content within lineages. So we had multiple individuals from multiple parts of the range, and we took an average for our venom complexity values. And for the diet studies, we kind of had to take what we could get, but many of them were museum specimens that represented collections from across the range of the species. And so we were getting sort of a dietary average value and a venom average value that we were relating here. Did that answer the question? Or give you more information to think about it anyway? Yeah. Okay. Other questions? Sure. In terms of regulatory evolution, we're just starting to really think about how sort of the optimization of what actually finds its way into the final venom mixture might occur. But there's only so much room in the gland. So if you express a bunch of some ineffective protein, or a protein that your prey has co-evolved maybe an effective resistance to, then that's just taking up space within that compositional space of just how much protein you can pack into that mixture. And so that you can imagine that being a maladaptive case for that particular focal gene that you're looking at, and where you might expect it to either evolve some new function or be deleted. And this sort of deletion addition just seems to be rampant across these clades. We haven't sequenced a genome yet that had the same copy number of any of these venom gene families. Yeah, Andre?
Have you been able to, I guess, identify novel venom gene families? Because I guess the method that we're using for kind of designating something as venom is based on knowing what's already designated as one, so you're going to get expansions and contractions.
Yeah.
So did you find kind of completely novel things?
So another main line of work within this Scales of Biodiversity project was to identify novel toxins, mainly through starting to sequence venom tissue and do proteomics on venoms from snakes that are much less well studied. This group is the best studied in terms of we know we've done proteomics has been done on almost all of these venoms, or some member within the clade, and then you can relate what's present in the actual venom mixture back to the genes that underlie it. So there is an effort in a lot of these traditionally considered non-venomous snakes, but they're actually rear-fanged and just non-toxic or not very toxic to humans. Colubrid snakes and dipsid snakes. So that effort is ongoing, and we expect to find novelty there. There was not an expectation for novelty here, and so to be honest, we really didn't even look for it. We kind of had our set that was based on sort of a diverse smattering of proteomes, and we worked backwards from that. Renan?
I was just curious, Matt, thinking mostly probably about the rodent part of the talk, have you found any evidence of loss or pseudogenization of the extra copies with, say, for example, reduction in the diversity in diet or something like that?
So in the case of both the snakes and the rodents, the sort of reconstructed ancestral phenotype is moderate. And so we're predicting evolution in both directions. There's no sort of increasing march towards more and more complexity here. And I didn't really point it out in the diagrams of the cytochrome P450 expansions, but there were all these green gene mark annotations, and then there were a bunch of yellow ones. All the yellows were pseudogenes. So there are several pseudogenes, including in the novel regions. So whether that is due to sort of incomplete sort of tandem duplication of the regions or subsequent pseudogenization, I can tell you that both things occur. Sometimes there's just a single frame shift that messes up the gene at exon 3, and sometimes there's only the back half of a gene present. Ivan?
So, Matt, there's evidence of hybridization across the rattlesnakes, multiple species, right?
Yep.
Are people looking at, or maybe you have looked at how genetic introgression, or if there's evidence of like introgression of venom genes across snake species? That's something that people have looked at.
Todd Casto's group at University of Texas Arlington is focused pretty heavily on that. They have an NSF grant with that exact idea in mind. So going to several hybrid zones and transecting them and looking at gene copy placement along these clines and things like that. Yep. Same with the wood rats. Marjorie's focus and sort of impetus for doing these is all 4 of these species actually hybridize at different contact zones. So a next follow-up there is also going to be to understand how these detox genes evolve and move across hybrid zones. 2 of the genomes were actually sequenced with a novel technology called trio binning, a bioinformatic method from a first-generation hybrid of 2 of the species. We got 2 genomes out of 1 hybrid. Okay. Thank you all.